welcome and thanks for coming to listen to who BBS nanotechnology is today. There we go. So who we are is a small 16-person biotech company. We're based in Europe, and unlike the talk we just heard, we have a similar burn rate, but we actually support about 3 times as many employees. We are an R&D shop, and what we're focused on is researching and developing the intellectual property of our chief scientist, who is on the faculty at the University of Debrecen in Hungary. I was brought on first as head of business and then as CEO when my project leadership was acknowledged. And what we're seeking to do is to bring the intellectual property of our CSO through the preclinical phase, develop IND-enabling packages, and then in a traditional licensing model, partner those technologies with pharmaceutical companies. It is, I think, the vision of many companies of our size to bring the technology through the clinic, but we are seeking to put one foot in front of the other. And my experience in the major pharma companies has shown me that the clinical development expertise is actually in the pharma companies, and I think that typically the pharma companies prefer to develop those products clinically themselves. So what we're seeking to do is to develop strong and convincing, compelling preclinical data packages and partner with the traditional pharma companies. We have a platform that is very broad and accommodates both therapeutic agents and imaging agents, and ultimately the vision actually connects with some of the talks that we're hearing at the plenary sessions next door regarding targeted personalized medicine, because what we believe we'll be able to do is to blend therapies with imaging and have the physician look in real time and determine what the prognosis and the progress and response of a patient is in real time. And that's, we call that, and others call that, theranostics. So I'll talk a little bit about, give you a little bit of information on where we are and what we do and what we hope to do. So we were founded in 2005, before I arrived. The proprietary nanoparticle delivery platform is called CytoCap. We're based in Hungary and also here in New York. We have 16 full-time employees. About half of them have PhDs. The others have bachelor's degrees. We are funded by venture capital money. We're funded primarily by Primus Capital Advisors, which is the preeminent VC tech firm in Hungary. And we're seeking a round C of investment, which will take us through the achievement of IND-enabling packages, and we think through the realization of partnering with pharma companies. So a little bit about the management team. There's me. I was a business development director at Pfizer. I was a BD director and a project team leader at Novartis. Started my career in industry as an R&D project team leader at Wyeth. Prior to that, I was a reviewer at the FDA for biologics and a program officer at the NIH. So I've seen the industry from soup to nuts, and hope to bring that experience to managing this small and very capable company. The founder and chief scientific officer is János Borbély. He was actually the Environmental Protection Agency director of the country of Hungary for 3 years, and during that time he realized that nanotech was going to be important in many fields, especially in biology, and he thought, you know, it would be really better if we were able to develop nanotech without releasing volatile organics into the environment. So he set about using his expertise to do this, and this is what he did. And the company is based on, as I said, his intellectual property. We've got 3 granted patents and about 15 patent applications in with the global patent authorities. So we can talk more about that a little bit later if anyone's interested. So we set about putting together a strong science advisory board, and we were lucky enough to get the Maxine Jochelson, who is here at the Breast and Imaging Center at Memorial Sloan Kettering in Manhattan. Leslie Botnick, who's the CMO of a very large radiotherapy practice that's based in LA but is actually countrywide. So we got 2 Americans and a couple of Europeans. We have Fabian Kissling, who's the director of the molecular imaging program at the University of Aachen, and the director of the Institute of Oncology at Debrecen, who has run a number of clinical trials, including phase 1 trials. So we have we believe we have the scientific expertise that we need to bring our program through preclinical IND enabling studies. So just a little bit about our operations. We operate out of a 5,000 square foot space in Debrecen, which is an area that's essentially the economy is fueled by the university and medical school, so there are a number of early startups and businesses related to the medical school in the area. As I said, we're focusing on IND enabling data packages that will allow us to partner with companies that are interested in clinical development in the US and EU. We think we have a good business model because we would like to continue doing discovery and innovation in Hungary. And this is a very efficient shop, and the scientists there not only are exquisitely precise in the development of this platform, but of course have the historical and practical understanding and the know-how to really develop these preclinical products very quickly and efficiently. We are seeking to validate our animal proof of concept either in the US or the EU. We're talking to some German, some American, and some Canadian companies about that. Similarly for PK/PD and toxicology. So any questions before I go on? Okay, good. I don't think I have to review the current problems in cancer therapy. Suffice it to say that nano and reformulation in general, I think, is a very interesting area. We've got a lot of good drugs, but they're sometimes more than a little bit too toxic, and a good nano product will certainly solve this. Of course, the first generation of nano encapsulation for cancer is very well established and has been quite successful, but there are a number of limitations with the technologies that are out there, and we and many others are seeking second generation nano encapsulation for cancer, and of course a safe, biocompatible, targeted approach is one of the approaches that should be tractable, and that's the approach that we're developing. So we call, as I said, our platform CytoCap. It uses second generation encapsulation techniques, and we're working on well understood cytotoxics precisely because they are well understood, and we believe strongly that we are starting to show a benefit over the traditional parent drugs. The release of the drug is targeted at the tumor site. And I think the interesting thing about our platform and our company is actually the chemistry. So we base our platform on well used, well understood, biocompatible, safe polymeric anions and cations that self-assemble. And there are a number of functional groups that are added to these polyanions before self-assembly takes place that allows us to, we have added a number of different targeting moieties and encapsulated a range of hydrophobic and hydrophilic payloads. So the flexibility and the precision and the great understanding with which the discovery group handles and develops these products, I think, is one of our important advantages. This is not a shotgun approach. This is a rifle approach, if you will. So what have we shown? And I'll show a little bit of data later. CytoCap versus the competing platforms in mice. What we have seen, and I think we've done xenografts. We've probably done close to 20 different xenograft experiments with a number of probably 10 different payloads. What we see on a very consistent basis is superior safety. And we believe that because we have seen superior safety in a number of different payloads with a number of different targeting moieties, that we will achieve the milestone and sort of the gold standard of a more favorable MTD. And that's what we're working on now. So what we see is increased overall survival and decreased weight loss. There are a number of other advantages, which I've alluded to. Self-assembly means simple manufacturing. Biocompatibility means safety. The manufacturing process is extremely straightforward and flexible. This is just a list of a number of the publicly acknowledged payloads and targeting moieties that we've used. We've submitted patents on a number of them. We also can, as I alluded to, encapsulate imaging agents, which can be licensed out as products on their own, but also can help us to realize the vision of nanotheranostics. So here is, I think, the one experiment that you probably want to see. Let's see. Whoop, wait a minute, okay. Right, so my pointer doesn't seem to be working, but the black is control, that's growing tumor. Red is naked drug, and then a number of versions of the nano-encapsulated payload compared with Calyx, which is the industry standard. It's in liposomal, it's like Doxil, but it's in Europe. But the important thing to look at is the dark blue DDS-PFA. It's given at half the concentration of Calyx and works almost as well. That's the targeted, nano-encapsulated, best version of the doxorubicin construct that we have. And as I alluded to briefly, weight loss is very favorable. And as I said, we're able to reproduce this data time and time again. So, you know, that's what our strength is. Imaging, we, compared with standard PET agent, we achieve 10% greater tumor uptake, but more important, 70% better tumor to muscle signal intensity. So, you know, we know our targeting's working, and this is a fabulous companion diagnostic at the very least. All right, I think that's about it. Finances, come talk to me. Any questions?
Thank you very much.
Okay, thank you.